I have a bit of a sore throat, so if I randomly start coughing, I'm really sorry. But yeah, so tropical lakes and human impacts on them. I know that most of you are no paleoecologists, some of you are, so I'm just going to start with a general introduction what paleoecology is, how it works, and then move on to my research. So what is paleoecology? It's the ecology of the past. Interactions between species have been going on for like a really long time, and by studying paleoecology, we can reconstruct past populations, landscapes, environments, and ecosystems, and sort of understand the changes that happened there like years ago. And by better understanding the changes, we can also better understand the drivers behind these changes. So climate is a huge driver, but over the past hundreds or thousands of years, depending on where you live, we humans also have a huge impact on our environment. So to study the past, we can look at written history, but not everywhere around the world you have a written history. It might not always be true. And if you go back for like 10,000 of years or even further back, there is no written history. So what we use then is a natural archive, and that can contain like fossilized biological remains or chemicals, and these can help us understand what happened in a certain area at a certain time. So how does this work? I'm going to use a lake as an example, as I work with lakes, so it's kind of easier to do that. So you have the lake, and then the area around it, that sort of the soil flows down towards the lake, is called the catchment area. And the soil contains a lot of information, and how fast the sediment is flowing towards the lake is kind of depending on a lot of things. So one of them is vegetation. So if you, for example, have a forest with loads of trees, trees have really big roots, and the soil sort of sticks to them. And trees get really old as well. So you expect a low sedimentation flow, especially compared to agricultural area. Quite often crops are like maize, they're annual, they have shallow roots. So you have a lot of disturbance on the land. Besides the vegetation, climate is also really influencing on like this sedimentation flow. So if there's like a really wet period, more soil is flowing down towards the lake compared to a drier climate for a few years. And all of the soil with the information we're interested in sort of accumulates here on the bottom of the lake and forms a natural archive. And it's not just the soil, it's also particles in the air that come down, fall into the lake and then sink to the bottom of the lake, but also everything that's living in the lake. So think about algae or fish, and at some point it dies and it sinks and becomes part of this natural archive. And then we come along and we take a little sample of this natural archive, a sediment core, and we analyze this in the lab. And for my PhD, I look at biological remains, 2 of them. The first being diatoms, and these are some examples of the diatoms I came across. They're single-celled algae. Their wall structure is made out of silica, so they're glass, and that gives them this really beautiful structure. They're great to look at. They're very sensitive to in-lake changes. So think about the change in temperature or the chemical composition of the lake, or even lake level. If there's a change in them, the composition of the diatoms will change as well. And by looking at the diatoms that sort of died and are not part of the sediment core, their composition can tell us something about what happened there, like what kind of change occurred. And they can be influenced by climate or humans. The other proxy I look at are pollen, and I recently got back into the lab and I started with the pollen slides. So these are a few photos of the first pollen I came across. I'm very excited about them. Pollen are reproductive cells of plants. Probably many of you know them through hay fever, but besides being really annoying, they can also be really beautiful. And they can tell us something about the vegetation change, like in the catchment area, so close to the lake. But some pollen, as for example the pine up here, they have air sacs and they can travel really far. So pine trees from like miles away from the lake, you can still find them in your sediments. Again, pollen can be influenced by climate or humans. So I get my sediment cores from Western Uganda, and indicated here with the red square, and it's a close-up of the area. This area is known for its high crater lake density. There are over 80 lakes here, and they can be freshwater, but also saline. And they can be divided in 4 clusters. And besides the high crater lake density, there's also a high population density, and this is growing. Most people living in this area are actually really depending on natural resources. So the lakes are really important for them for like the livelihood, but also for an income, because they're really beautiful and a lot of tourists actually come there. But a growing population means that they're also putting a lot of strain on these lakes, and loads of the lakes are heavily impacted by humans. For my PhD, I focused on the most southern cluster, the Bunyaraguba region, again, close up. So I took 6 sediment cores of 5 lakes, of which 4 of them are in impacted area. So that's Katinda, Mirembe, Karia, and Kabaroji. And Lake Kamanzuko is in a relatively pristine catchment area. But we also have data from previous research on lakes from this area as well. So with this, I'm trying to answer the questions: how have these lakes changed over the past 50 to 150 years? And how have humans influenced these changes? Well, to show some of my results, I'm going to focus on Lake Katinda and Lake Mirembe. At the moment, I'm writing a paper on them, so it's kind of easier to focus on them. This is Lake Katinda, this is Lake Mirembe, and as you can see, they're not like 30 meters apart from each other. So if there's like a change in climate, you would expect that the change will have occurred like in both lakes. They're both affected by that. So first, Lake Katinda. And this is a diatom diagram. Again, assuming that most of you have never seen this before, I'm just going to talk you through it. So over here we have the depth of the core. So this core was around 26 centimeters long. Normally you have the date here as well, but due to COVID, we haven't got our dating back yet. But there's one guess that like the bottom of this core, around 26 centimeters, would be around 1965. This is organic matter and the carbonates in the core. These are some of the diatoms. So in total, I counted over 80 species from this core. But I'm just showing here the most dominant species, so the ones occurring at 3% or more. This is the concentration of the diatoms, and here we have the diversity. And we have everything sort of, we have zones, so there are like 3 big zones and 1 subzone, and all of them are significantly different from each other based on the diatom composition. And this extra graph shows where the diatoms, what their habitat is, where do they live. So by reading this, we're starting at the bottom, so zone 1, and let's say that this is from 1965 to 1985. And based on the diatoms, compared to the lake nowadays, it had an average lake level, and a large quantity of the diatoms are epiphytes. So that means they live on macrophytes, which are water plants, or like green algae, where they actually attach themselves to. So what you expect is a slightly greener lake and maybe some more plants. Moving up in time, we go to zone 2, and there's, you can see these are slightly disappearing, and at the end, these 3 over here are increasing, and that's like the part of the purple ones of the aerophilic diatoms. These live outside of the water body, so an increase of them could indicate land, well, disturbance on the land. So it could mean that there is a higher influx of sediment coming from the land into the lake. And based on the other diatoms, it looked like it had a relatively low lake level. Zone 3A, even though it has a peak here in these aerophilic diatoms, it sort of settles. So it looks like the sediment inflow is slightly less. And based on the other diatoms, lake level rose again. So we think it's a higher lake level. And the most modern data that goes up to 2018 when we took this core, there's a slight increase of epiphytes again, and we think it's an average lake level, and then with more green algae in it, which makes sense because when we actually took the core, the lake was very green and looked very impacted. So makes sense. Moving on to its twin lake, Lake Marembe. Again, the diatom diagram. I'm going to talk you through this as well. This lake, the core of this lake was a little bit longer. It's almost 60 centimeters long. Again, we don't have the dating, but it looks like it might be around 150 years old. So in the first zone, zone 1A, it looks like sort of an average lake level, but an increase of evaporation. So it might have become slightly warmer or something else was going on. Zone 1B has a peak here of these diatoms, which sort of prefer more saline water. And based on the other diatoms, it looked like a huge drop of lake level. So this could mean an increase of evaporation. So a warmer period. Zone 2, slight increase of lake level again. There's a high percentage of epiphytes, so like Lake Catinda, we think it might have been much greener or more plants living near it. And the most, the top zone, the most modern one, going up to 2017 when we took the core, looks like there's an increase of precipitation and lake level overall have risen a little bit. So what would have been great if, or the goal eventually of this is to compare these lakes to each other. However, we need the dates for that and we don't have that yet. So it looks, based on the organic matter, that all 4 zones from Lake Catinda fit in the most top zone from Lake Marembe. And when we have the dating, we could sort of divide this zone into the exactly same dates as these 4 zones from Lake Catinda and then compare and see what might have been a human influence or impact on this lake. And yeah, that's it. Any questions?